Later, we have Juliette give us the topic of impacts and drivers of signal crayfish population in UK river systems.
Thank you. What do I do? Okay, well, it works. So, hi everyone. So today, yeah, I can lower it. Much better. I'm gonna present to you guys my bachelor's thesis, which is about impacts and drivers of signal crayfish, Pachyphistachus lenusculus populations in UK rivers. So today I'm gonna talk to you guys about this little ugly animal, and this is the signal crayfish, so Pachyphistachus lenusculus again. So it's a freshwater crustacean species that is native to North America, in particular to the United States of America, and it was introduced in Europe in 1960s, and it has become an invasive species ever since then. So it negatively affects the freshwater ecosystems of Europe by reducing the local macroinvertebrate communities, since it heavily preys and feeds on them, and then it furthermore alters the freshwater ecosystems by intensively feeding on plants, fish, and benthos, zoobenthos. They eat everything, basically. And then furthermore, they also are responsible for the decline of the endemic crayfish species to the British Isles, which is the white-clawed crayfish, because it outcompetes them. So the problems that were the main basis of my research were that the species was introduced in the United Kingdom in the late 1970s. And ever since then, it spreaded and it well established in the wild across the whole country. So several populations have been documented across the United Kingdom. However, there was little knowledge about its presence and its ecological impact in the east of England. Okay, so the aims of this project were to assess the presence of the species in the east of England, in this case in the county of Essex, and so to assess its impact on the local macroinvertebrate community and to see if there was any link between the signal crayfish population and water quality, in this case taking into account phosphate and nitrate. So for our hypothesis, we expected to find less macroinvertebrates and less macroinvertebrates species in waters with large crayfish populations, and to find more signal crayfish in rivers with lower levels of nitrate and phosphate. So the study took place in the county of Essex, east of England, and we surveyed 3 rivers, so Salary Brook, the Stour, and Layer Brook. To talk briefly about the methodology. So to assess the signal crayfish abundance in every river, I will place 10 of these traps. So these are called funnel traps with some bait to attract them. I put bacon, actually. And the point of these traps is that, as you can see, the yellow entrances, they allow the crayfish to go in, but not to go out. So every day for a week, I will check on the traps, and any specimen that I would have collected through the trap, I will bring them back to the lab to count them and to identify their sex and their age in order to get a population size estimation and structure. And to assess the macroinvertebrate species richness and abundance. So as used what is called a surface sampler, as you can see in this picture. And the point of a surface sampler is to disturb the river substrate that is inside the square, and any organism, microinvertebrate, will flow inside to the net. And then I will collect the microinvertebrates from the net and bring them to the lab to identify them via a microscope and to count them. Then for water quality, so I collect a water sample with syringes, and then I transfer them to a test tube with 0.2 filter, as you can see in the picture, in order to get the samples as pure as possible. And then I will bring them to the lab, where they would undergo through a ion chromatography test in order to get the values, the levels of nitrate and phosphate. And then to test all of our hypotheses, we used a one-way ANOVA statistical test. So what did we get from the study? So the study confirmed the presence of the species in all of the 3 rivers. And as you can see, the Stour was the one with the largest populations, almost 300 individuals. followed by Layer Brook with 39, and then unfortunately I only got 2 crayfish in Salary Brook. Microinvertebrate species richness and abundance. So the Stour was the river with the most microinvertebrates as well, the most microinvertebrates and the most species. As you can see here, 6 in the Stour, 5 in Salary Brook, and then only 4 in Layer Brook. So we found several taxa, like freshwater crustaceans, freshwater shrimps, snails, and so on. And so the ANOVA test showed that there was a correlation between the crayfish abundance and the microinvertebrate abundance and species richness only in Layer Brook, contrary to what we expected. And then, in regards to water quality, so our hypothesis in regards to nitrate was wrong, so there was no correlation between nitrate levels and crayfish abundance. However, we found a correlation between phosphate values and crayfish abundance only in the Stour, though. So how can we comment these results? For example, when I surveyed Salary Brook, heavy rainfalls and currents took place. And Salary Brook is a very small, narrow stream, so it took away almost all of the crayfish, so I only found 2 there. It's also worth to mention that the surveys took place in late autumn, and in that time, actually, the species starts to hibernate underground in the river substrate to prepare itself for winter. And furthermore, there is no record of the species introduction in either Salary Brook and Layer Brook, and that, for example, could help us to understand the growth and the evolution of the population throughout time. But for example, in the Stour, so there is previous evidence from the 90s, which could furthermore explain the large population that we found over there. And more surveys should occur in summertime because that's when the species is the most active. So in this case, we will have more accurate results as well. So to discuss microinvertebrate species richness and abundance. So the life cycle of a species can affect its distribution and abundance in a given time of the year. For example, many species have only found them in a larval stage, for example. It's quite interesting that we found the lowest taxa number and microinvertebrates number with a large crayfish populations in Layer Brook. More sampling and surveys should occur in order to explain this aspect. And for example, in the Stour that I found such a big species, I was expecting to find a correlation, between the crayfish and the microinvertebrates. However, there is some research that says that, for example, very large crayfish populations can actually have less impact on the microinvertebrates because there is more competition within the signal crayfish and more recurrent episodes of cannibalism because they eat everything, they're awful. So, to discuss water quality. So the correlation that we found between phosphate and crayfish abundance in the Stour could perhaps suggest us the most adequate phosphate values for the species. But however, more sampling would need to take place in order to confirm this idea. It's also important to mention that next to the Stour and Layer Brook, there are farms which release nitrogenous discharges into the rivers, which negatively affect for the species. They don't like high levels of nitrate. More sampling efforts, perhaps on a seasonal basis, could help to explain any connection, and especially the one that we found in the Stour. So, conclusions of this study. So, the study confirmed the presence of the species in Essex. As I mentioned before, seasonal surveys need to happen in order for a better understanding of both the macroinvertebrate and the signal crayfish dynamics, and it provides sites that need monitoring for the species in the county of Essex, which are actually currently monitored for the species. And so these are the people I would like to thank that helped me in my research, and thank you very much to you all and my references. Thank you.